Okay, so this time we're going to be discussing about fatty acid metabolism. Now, I know some of you have already viewed the PowerPoint that I have included for this discussion. Wait lang, naman kulit, tatlong pusa na nagkakagulo. Okay, so I'll be updating it because I added one more metabolism, which is steroid metabolism. Okay, so when you talk about fatty acid metabolism, okay, so where is my control? Okay. So always remember when you talk about food digestions the metabolism per se. Remember we talked about and it's divided between anabolism and metabolism, and usually you have your digestive system that would aid in this process. Now, take note, we know that the triglycerides are a large molecules, and unlike carbohydrates and proteins, they are not water soluble. So, since they are not water soluble, they tend to cluster in large droplets, which, when they're um, in a watery environment like a digestive tract, and then the digestive process needs to break down. Shh, please come on, Jen. Right. The digestive tract needs to break down those large droplets so into smaller droplets and then enzymatically digest it. Okay, so you call that enzyme that digests it as lipases. Okay, that's the name of the enzyme. So take note. Now, always remember that the mouth plays a mechanical role in breaking down the food, and as far as lipid digestion, halos konti lang siya. Okay, hindi niya kaya. Unlike if you're gonna compare it with starch, wherein starch, automatically nagkakaroon na yan ng amylase action. Kaya minsan pag naglagay kayo ng, alam mo yun, isang maliit na tinapay sa bibig ninyo, later on, halos, na siya, watery na siya kasi nadadigest na siya. Okay, so this time before we go to the different pathways, we're gonna be reviewing the different areas of digestions of lipids particularly. So let's start with the digestion in the mouth. Now, usually, as I mentioned earlier, it is the first entryway for the digestion of lipids. So we know that we chew, we masticate the food that we eat, we break it down mechanically using our teeth and our tongue. And the type of lipase that is available in our mouth is what we call the lingual lipase. So lingua meaning the tongue. So it's inside our mouth, and it starts the enzymatic action, but it's not enough. It's not strong enough to um break it down further. Okay. So yeah. So usually it tends to continue further into the stomach. So in the stomach, there is the churning of the stomach muscles mixing with different food particles and fat. Now usually the cells of the stomach also has what we call the gastric lipase. Okay. And it's adding more enzymatic action. Now, yung lingual lipase that was available from the mouth will still be active in the stomach. So they will add more role, or they will play a minor role in the digestion. Usually, the major enzymatic action will be happening inside the intestine. Okay? So, as I mentioned earlier, so sa intestine na tayo. So yan, so kanina sa mouth, now we are in the stomach, then into the intestine. So inside the intestine, we have their different materials or chemicals that tend to help in the digestion. So unang-una, so bile. So bile is actually, take note that it is made, let me just open my pointer. So it is made in the liver, and it is stored in the gallbladder and released in the duodenum, in the first section of the small intestine. So, di ba, you have, uh, so always take note where it is actually stored and where it is created. Okay. So you also have the bile salts are what also we could consider as hydrophobic and hydrophilic side. Okay, that's why it tends to interact with water and fats. Now, you can also inside the bile you can also find micelles of lecithin, cholesterol, bile salts, and inorganic ions and pigment. Kaya nga 'di ba, pag nakita kayo ng bile, it's usually dark green color siya. Yan. And it actually acts as an emulsifier also. So when we say emulsifier, it breaks down the large fat globules into smaller droplets. Okay, so kunyari kumain kayo for those who are eating pork o kaya yung sa, masarap yung ano eh, sorry ha, kung hindi pa kayo nag-breakfast, pasensya na, hindi ko sinasadya, nagkataon lang sa discussion natin since we're talking about digestion, 'di ba? For those who eats tocino, 'di ba? Yung masarap yung ano eh, yung konting fat ng tocino. Okay? So yan. So yung kinagat ni Don, it's gonna be broken down to smaller components, which is gonna be causing larger surface area. 'Di ba? Remember to take note that pagdating sa interaction ng mga chemicals, the greater the surface areas exposed, is as in chemistry 1, the faster the interaction is. So, ganun din po sa fat. So, kung mas malaki yung fat, kaya di ba pag nagluluto tayo, mas maganda pag yung niluluto mo is cut into smaller pieces. Diba? Para mas mabilis yung pagluluto. Ganon din sa pagdating sa lipids. If it is smaller pieces, it tends to act faster. Okay? So, the Okay. So when I say, so going back, so as I mentioned earlier, since it has already emulsified it, so nag-increase yung areas for action. Now we also have the pancreatic lipases. So this is actually coming from the pancreas and it is sent to the small intestines for glyceride digestion. So the glyceride are broken into fatty acids, monoglycerides. So yung triglycerides natin, maghihiwalay sila. Okay? And some free glycerol. Then you also have cholesterol and fat-soluble vitamins that need not be enzymatically digested. Okay? So yeah. So here, okay. Now, the products of the fat digestions like fatty acids, monoglycerides, glycerol, cholesterol, and fat-soluble vitamins need to enter the circulation so that they can be used by the body. Now, take note that bile helps in this process, particularly bile salts, which cluster around these materials for fat digestion to form structures called micelle. Kasi po, once the fat cannot pass through the barrier, so it has to form a micelle. I think we already discussed this when we discussed about lipids in the past. Okay, so this is actually allows those fat materials to be able to be absorbed by the microvilli of the small intestines. Okay, so once it's already diffused into the membrane, once it enters the intestinal cells, it is already broken back to its forms, and then yung bile salts will be shifted back for further reabsorption ng iba pang lipids. Okay? Now, so medyo mahaba yung sequence ito. So once inside the intestinal cells, so ito ngayon. So, di ba binreakdown natin siya itong monoglycerides, glycerol, and fatty acids. This time, we're gonna be bringing them back together to form now your monoglycerides and then other fatty acids and other lipids. And then it's gonna be transported into the bloodstream. Okay? And including fat-soluble vitamins. Now, yung long chain naman na fatty acids, they would be forming now the triglycerides within the intestinal cells with cholesterol and fat-soluble vitamins. And then, papasok sila ngayon into transport by what we call the chylomicrons. Okay? So in this slide, you're actually seeing here the steps of how it is happening. So unang-una, you have your emulsification of your droplets into smaller ones. Okay? And then once it's already been emulsified, it's smaller pieces already, and then it's broken down into different fatty acids and monoglycerides by enzymatic pancreatic enzymes. And then that's the time it's going to be able to enter. Wait lang guys. Yes? Did somebody raise their hand? Or napindot lang po? Okay, sige. I guess napindot lang. Okay, let me just go back here. Yeah. So yeah. Now once inside, okay, so it will now be forming back into triglycerides, and then your sorry, not your carbohydrates, your short and medium chain fatty acids will be absorbed by the capillary, although it's not shown here. But your long chain, yung mga monosaccharide and other monoglycerides, will form your triglycerides, which will be joining now your chylomicrons, and it will enter now the lymphatic system. Okay? So, yan po. Okay, so that is for the digestions. Now, take note, including din po also dito to forming the chylomicrons as it passes through the barrier for the microvilli in the small intestines, yung cholesterol na nadadid na kinakain natin. So, if in case we ate, let's say, corned beef, for those who eat corned beef, meron po kasi yung mismong lipids or yung fats na kasama po dun sa corned beef mismo contains cholesterol. So, kasama po siyang mabe-breakdown po siya dyan. Will be, this is also the same way it is gonna be absorbed by the body. Okay? So, any question regards with metabolism before we proceed to fatty acid degradation? Question, guys?
Wala
Wala
naman.
naman po.
Okay. Okay. Let's continue. So next is what we call the fatty acid degradation. Okay. Before we proceed with the degradation, let's have a few backgrounds how this was actually introduced or how was this found out or na discover. So you have there yung German biochemist Franz Knupp. So actually he devised a labeling experiment on radioactive tracers. So they have there, he had his dog ingest or eat fatty acids that were labeled with methyl groups and the phenyl group na attached to it. So ito yung mga mismong materials na doon. So what he did is was able to isolate the metabolized fatty acids from the urine of the dogs. And he found that phenyl acetate was formed when the fatty acid is even number. Now take note that this is very important kasi po, this is actually gonna show you what your products is gonna be when you have odd and even fatty acids. Okay? So pag even numbered is you're gonna be having phenyl acetate, ito po, and then acetate. And then if you're having, sorry, yeah. And then for if it's odd number, it's benzoate and then acetate. Okay, so yeah. Now, let's go this time with the actual steps in fatty acid oxidation. So, it's actually an aerobic process which is breaking down fatty acids into acetyl coenzyme A. Now, the fatty acid move in the pathway as CoA derivatives utilizing NAD and FAD. Now, at the same time, these fatty acids are activated before oxidation, utilizing ATP in the presence of CoASH, CoA thiol, and acetyl coenzyme A synthase. Now, before long chain acyl coenzyme A enters mitochondria bound to carnitine. Now, inside the mitochondria, beta oxidation of fatty acid takes place in which two carbons atoms are removed to form acetyl coenzyme A from acyl coenzyme A at the carbonyl end. Okay, now the bond is broken on the second carbon or the beta carbon. So from the terminal, it's the second carbon. That's why it's called a beta oxidation. Okay. So that is the reason why it is called beta oxidation. Now this actually provides energy from fats. So let's actually go to the steps. Now take note guys, the way I presented the steps here is medyo compact po siya pagdating sa first kasi meron po akong gustong i-point out dito. We have what we call the carnitine shuffle which we'll be explaining here on the next three slides. Now the carnitine shuffle actually is what's causing yung pag-move ng mga fatty acids from one location in the cell. Okay? Now, if you would be looking into Lippincott biochemistry, yan po yung mga sinasabi niya po. Pero yung iba po, if it's gonna be a lighter discussion of biochemistry, hindi na po nila dinidiscuss yun. Kaya lang, since you're a nursing major, um, it's it's a plus for you that you'll be able to understand this as well. Okay, so yeah. Now, so the first one in the um, fatty acid beta oxidation is the activation reaction. So this actually results in the production of fatty acyl coenzyme A. Okay, and the thiester bond is formed between coenzyme A and the fatty acid. Now it requires energy in the form of ATP. So coenzyme A is also required in this step, and the product of fatty acyl coenzyme A has a high energy thiester bond between the the fatty acid and coenzyme A, and then the enzyme is acyl coenzyme A ligase. Now, take note that this is happening in the outer membrane of the mitochondria. Okay? So, nasa outer membrane siya ng mitochondria. Okay. Still on step one, so this is actually showing the reaction. So the mechanism of the fatty acid coenzyme A in the mitochondrial matrix involves a carrier molecule called L-carnitine. Kaya nga, di ba guys, I'm not sure if you've heard this. Ito yung actually kasama dun sa mga, for those who are interested in all this dieting, yung lagi na may mention, I think it's also included in Dole, I think it's Dole, yung pampapaya, ano nila, yung shape up yata yun. I forgot the name of that juice drink na may L-carnitine siya. Yan. Because that is actually what they're trying to achieve, that this is actually gonna help in the shifting of your fatty acids inside your cells. Okay? So yan, pag sinabi na may L-carnitine, nasa utak talaga nila is for papayat daw yan. Okay? Yun ang sinasabi dyan. Pero you'll understand this later on. Okay? Kayo na maghusga. If you really need to drink those things or just exercise and eat properly and burn those excess food that you eat, yun lang. Okay, so the first step is catalyze the enzyme called carnityl acyl transferase 1, which is the transfer of fatty acyl group to L-carnitine, producing acyl carnitine and coenzyme A. So, yan po yung nangyayari dyan. Okay, and then the carrier protein located on the mitochondrial inner membrane transfer the acyl carnitine into the mitochondrial matrix. So, if you're going to look here, okay, so it's actually being entered now into the inside the mitochondrial membrane, and then the acyl transferase 2 catalyzes the regeneration of fatty acids, which now becomes involved in fatty acid beta oxidation. Okay, so if you're going to look here, yeah, okay. So you have your acyl carnitine, pasok, 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 and then tanggal si acyl coenzyme A, carnitine is shifted up. Ito po yung mismong carnitine shuffle na tinatawag. Okay? So moving out, in and out of the mitochondrial space. Okay. Step 2. So the reaction is an oxidation reaction that removes a pair of hydrogen from the fatty acid. So it will actually produce FAD to produce FADH2. This hydrogenation reaction is catalyzed by the enzyme acyl CoA dehydrogenase and results in the formation of carbon-carbon double bond. Okay, so this is it's a formation of a double bond, and the phosphorylation yields 2 ATP molecules for each molecule of FADH2. Okay, so this is actually what's going to happen. So here, you have here your beta. Ito po yung beta natin. Okay, so the beta now will have a double bond with the omega. The gamma, sorry, the gamma. Okay, now the third reaction involves the hydration of the double bond produced in reaction 2, and as a result, a beta carbon is hydroxylated. Okay, and this reaction is catalyzed by the enzyme enol coenzyme A hydrase. Okay, so here, if you will notice, since may double bond tayo dito, okay, it's now going to have water added onto it. So, there is a rule in um, organic chemistry, which is what we call the Makornikov's rule, wherein yung carbon na mas malapit sa dulo, siya yung unang tatanggap ng excess hydrogen. Okay? So here, so ito ngayon, nagkaroon siya ng CH2, at siya naman yung tumanggap ng extra hydroxide na na-produce from the water. Okay? So this is Markovnikov's rule. Don't worry, hindi ko siya sinabi dun sa, hindi ko siya sinama sa exam, pero usually guys, sa organic chemistry, we have the Markovnikov's rule na yung sino yung nasa dulo, siya yung unang tatanggap ng hydrogen. And then, yung subsequent na kasama ng double bond, then, siya yung tatanggap ng remaining component na nandiyan. So, if in case you have, let's say, hydrogen sulfide, the hydrogen will be attached to this carbon, and then the sulfur will be adding here, which is the same with hydroxide. Okay. Now the fourth step here, so in this oxidation reaction, the hydroxyl group of the beta carbon is now dehydrogenated. NAD1 is reduced to NADH, which is going to produce 3 ATP molecules. So do you think though that this is happening inside the mitochondria? So the numbers are different. So NADH produces 3 ATP, FADH2 produces 2. Pero if it's in the cytoplasm, it's gonna be minus 1. Okay? So here, what will happen now is you're gonna be having a carbonyl. Okay? And then once you have already this is the final reaction. Okay? So the final reaction catalyzed by the enzyme thiolase, and that is cleaved releasing down your acetyl coenzyme A. So this is the process is thiolysis, wherein the coenzyme A will attach itself on the beta carbon, okay, here, and then it will now form your acetyl coenzyme A. Now, the shortened fatty acyl coenzyme A is further oxidized by cycling through sections 2 to 5 until the until the fatty acid carbon chain is completely degraded. So the fatty acid coenzyme A is produced by beta oxidation of fatty acid that enters the citric acid cycle. Okay? So yung products ito, diretso siya sa citric acid. Okay, let me see. Wala ba dito yung ano ko? Ay, wait. Ito muna. So, this is actually the mechanism. Hindi siya. Wait lang guys ha. Ayan. Wala. Ayan. So, if you will notice here, this is your FAD now, entering the system. Teka lang guys ha. May nag-raise ng hand or wala? Ayan. Meron lang siyang sound eh. Hindi ko pa na-edit na tanggalin yung audio eh. So, yan. So, this is your hydrate step 3. And then, so slowly siya hanggang sa maubos niya siya. So, then the step 4, yan. So, nag-carbonate na siya. And then, you have your coenzyme A now attaching itself. Yan. Okay. Now, this is now the new set of alpha and beta carbons. And then you have one product of acetyl coenzyme A. So, yan. So, repeat ulit hanggang sa makonsume lahat. So, step 1, FAD action. Step 2, tama. And then that's step 2, 3, step 4, and then step 5. Now, unfortunately, guys, yung website na kung saan makikita to, I'd love to give it to you, but since hindi na po sinusupport yung shockwave since 2020, wala na po siya. So, I tried going back to the website, wala na eh. As in, hindi na nila in-update. Okay? So, I was able to get this a couple of years ago, screen record ko na lang, kaya pasensya na kung may audio siya. Okay? So if you will notice, you have already 3 sets. This is already the 4th cycle. So 2, 3, 4, and 5. So this is actually the last cycle kasi po ubos na yung inyong carbon molecules here. So this is the last cycle. Ayan. Okay. So that is how your beta oxidation is happening. Just go back to PowerPoint. Now, take note, I have included some key things to remember. So, if it's even numbered, the beta oxidation ends with 4 carbon acyl coenzyme A, which is broken down into 2 acetyl coenzyme A units. So, wala kang add walang problema, all of them will become acetyl coenzyme A since it's even. And then all of them will enter the citric acid cycle. But if you have odd number as acyl coenzyme A, beta oxidation also happens, but the problem is that the difference, instead of having all acetyl coenzyme A, one of them is going to be propionyl coenzyme A. So propionyl is 3 carbons. Okay? So yun po. So yun yung pagkakaiba. So just simple logic. So if it's even, all of them will be acetyl coenzyme A, and if it's odd, meron ka isang propionyl. So if you're going to look into the overall reaction of a beta oxidation of palmitate, or usually in any fatty acid, so you have there C acyl coenzyme A, so that is the number of carbons. So let's say you have carbon 18, therefore it's going to be having C N16, acyl coenzyme A, FADH2, NADH, hydrogen ions, and acetyl coenzyme A. Okay, so that's the overall reaction for fatty acid degradation. Okay, now if you're going to look into this further, you might see other names depending on some resources are saying on based on the different location. Pero let's just call it na lang as beta oxidation. Okay? Kasi yun lang naman din yung sequence niya. Now, if you're going to talk about the overall computation of ATP molecules that we can find in just one palmitate, okay, so start with palmitic acid. It's broken down to form palmityl coenzyme A. Now, it's now going to be broken down via beta oxidation to form now your 8-acetyl coenzyme A. Okay. Now take note that the 8 acetyl coenzyme A during fatty acid degradation or beta oxidation, you produce 7 FADH. You have also 7 NADH. Okay? And then, since sabi ko nga kanina, this is times 2 or this is equivalent to 2 ATP. Remember when we talk about electron transport chain? Yung FADH produces 2 ATPs while NADH gives you 3 ATPs on each consumption. And then, ito namang 8 mong acetyl coenzyme A would be making the citric acid cycle turn 8 times. So, di ba, kung dito you have 3 NADH, isang FADH, and isang ATP, so times 8 mo lang lahat yan. So, that's 72, 16, and then 8. So, overall, you have 131 ATP na napoproduce from the fat. And then, since gumamit ka ng ATP during the reactions here particularly, yan, therefore, your net production is 129 ATPs. Okay? So, yan po. So, that is actually the reason why you can get a lot of energy in terms of fat. Okay? That's almost times 2 in terms of carbohydrate, times 4 in terms of carbohydrate capacity. Okay? Any questions so far before we proceed, guys?
None po, ma'am.
Okay. Thank you. So, let's continue. Teka lang guys ha. Wait lang. May nanggugulo. Wait lang. 1 to 2 minutes. Okay. So, let's go back. So, okay. Okay, so this time we're going to be talking about ketone bodies, right? So, for acetyl coenzyme A that is produced in beta oxidation of fatty acids to efficiently enter the citric acid cycle, make sure that there is an adequate amount of oxaloacetate. Now, if in case glycolysis and beta oxidations occurring at the same rate, so definitely there will be a steady supply of pyruvate from glycolysis that can be converted to oxaloacetate. Now, if the oxaloacetate is too low to allow the acetyl coenzyme A to enter the citric acid cycle, acetyl coenzyme A is converted to what we call the ketone body. So, ito yung tatlong ketone bodies natin. So, you have beta hydroxybutyrate, acetone, and acetoacetate. Okay? Okay. So, things to remember when you talk about ketone bodies, you have what we call ketosis. Actually, this is a metabolic rate characterized by elevated levels of ketone bodies in the blood and urine. So, ito yung actually favorite ng mga nagkiketo diet. Okay? Kasi nga po, parang mas bumibilis daw yung mismong metabolism nila in that process. Okay? Then you also have what we call physiologic ketosis. So this is a normal response to low glucose availability, such as low carbohydrate diets and or fasting that provides additional energy source for the brain in the form of ketones. Now, during physiologic ketosis, ketones in the blood are elevated above the baseline levels, but the body acid homeostasis is maintained. So hindi pa po nagbabago yung overall body pH natin. Medyo mataas lang, not exactly mataas. Remember, we're talking about pH. So ketones usually causes it to become acidic. So yung blood medyo mas acidic, so pH would be lower, but it's not changing the whole body pH level. Okay. Now, the other one is ketogenesis. So, ketogenesis is actually a process wherein we produce fatty acids from amino acids that are ketogenic. So, when you say ketogenic, these are amino acids that can be converted into form fatty acids. Okay. Now, our bodies continuously produce ketone bodies in low amounts, but in certain cases, like starving, when carbohydrates are present in less amount in diet, ketogenesis is preferred to compensate for energy component requirements. Okay. Now, ketogenesis occurs continuously in a healthy individual, but under certain conditions, when there is increased concentration of fatty acids or carbohydrate reserves are decreased, ketogenesis happens at a higher rate. So this is actually for those who are interested in keto diet, ito po yung gusto nilang ma-achieve. Okay? So under low blood glucose level, usually in starving, what would happen? This is what's gonna happen. Or pag sobrang pagod na, or sobrang ubos na yung glycogen natin. And there is insufficient insulin, particularly those who have type 1 diabetes. Okay, so this is usually what's gonna trigger your ketogenesis. Okay. Now, all the main body parts such as the brain, skeletal muscles, heart can utilize energy from ketogenesis. And insufficient gluconeogenesis results in hypoglycemia and excessive production of ketone bodies resulting in fatal conditions called ketoacidosis. Okay? So yeah. Now let's go now to the different steps of ketogenesis. So the first one is so take note, by the way, before I proceed, that the ketogenesis occurs in the mitochondria of liver cells. So there actually, if I'm going to look at general on it, there is one, two, three, four steps lang yung ketogenic steps. Okay, so unang-una po is yung acetyl coenzyme A is condensed. So dalawang acetyl coenzyme A is combined to form your acetoacetyl coenzyme A. Okay? Teka lang guys. Hindi pa yun. Parang i-off ko muna yung ko. Nag-a-alarm kasi yung ko pala. Yung Facebook pala yung tumutunog. Sorry guys, akala ko kayo. Sige, in-off ko muna yung yung Facebook. Kaya ko na lang open ulit sa isang browser. Okay, so yeah. So the first one is with the enzyme thiolase, you combine your two acetyl coenzyme A to form your acetoacetyl coenzyme A. Okay. So the second one is that you're gonna catalyze beta hydroxy beta methylbutyryl coenzyme A synthase, or what we call HMG CoA synthase, yun yung shorten niya, and uses water to add another molecule of acetyl coenzyme A to the beta carbon of acetyl coenzyme A. So dadagdagan niya yun siya. Okay? So this step produces beta hydroxy methylbutyryl coenzyme A, which is branch 6 carbon compound and intermediate for the synthesis of cholesterol in cytosol. So here actually we will see this again later on when we talk about cholesterol synthesis. Okay? Now third step, okay, so acetyl coenzyme A is cleaved by HMG-CoA lyase, producing acetoacetate. Okay, so yun po. And then the last one is, given that it's already in the acetoacetate, it can now either be uh, go to two steps or two products ang pwede niya i-produce. Pwede siya maging acetone or maging beta, D-beta hydroxybutyrate. Okay, so yung dalawa po yung kanya pwedeng pagdaanan. Okay. Now, what is it that ketogenesis is important? Because ketogenesis is used to get energy by the brain, heart, and skeletal muscles under fasting conditions. Kaya nga di ba, kahit nagutom kayo, you're still able to work because of the ketogenesis. Second, ketogenic diet, which is low carb, fat rich diet, is used these days to lose weight. Yung sinasabi ko sa inyo kanina. The idea is to utilize excess fat stored in the body to get energy, but excess ketone bodies can lead to various complications in ketoacidosis. Kaya usually guys, minomonitor to. I had a friend, a close friend that underwent a ketogenic diet talaga. As in, to the point na very scrutinizing siya. Talagang binabasa niya yung labels, chine-check niya ano ba usually nandoon, pati yung level of carbohydrates kasi nga gusto niyang ma-prove ano yung ma-start yung ketogenic sa kanya. Well, the good thing about it on what I've seen with her, naging maganda naman yung result. Yun nga lang you really have to make sure that you monitor your sugar ay your ketone level sa blood mo kasi nga it can lead to ketoacidosis, which is something that you need to avoid. Okay? Pero yan, majority ng mga nag-keto diet, maganda siya. Tapos, even the coffee guys, yung bullet coffee, yung nilalagyan ng butter pa nga. Yan, yun yung kanilang, wala silang sugar, yun yung dinadagdag nila. Okay? Now, in ketoacidosis condition, the kidney excretes extra ketone bodies with water resulting in fluid loss. Okay, so kaya actually parang water loss lang siya sa katawan. And then, diabetic patients are greatly affected by ketoacidosis because insulin hormone is the main regulator of the process. Okay. And then also, symptoms of ketoacidosis include frequent urination, breath smelling like fruits or acetone, nausea, shortness of breath, fatigue, excessive thirst. Okay? Now, the level of ketone bodies present in the body can be tested rather by blood serum or urine sample analysis. Okay? So, yan yung sa ketone bodies. Any question regarding ketone bodies and ketogenesis before we proceed to the next topic, to the next component or next pathway?
Wala naman po, ma'am.
Okay. So let's go to the next one, fatty acid biosynthesis. Now take note that this process is combining 8 2-carbon fragments of acyl groups from acetyl coenzyme A to form your 16-carbon of saturated fatty acid or your palmitate. Okay. Now, palmitate can be modified to give rise to other fatty acids. Now, take note of the modification. It includes chain elongation to give you longer fatty acids like 18 carbon stearate, desaturation, giving unsaturated fatty acids. And then here is the formula for fatty acid biosynthesis. So you have acetyl coenzyme A, 7 rounds of alanyl coenzyme A, give and 40 NAD and 14 hydrogen ions, giving you palmitic acid, 7 carbon dioxide, 8, wait, sorry, hindi ko nasa, 8 coenzyme A, 14 NADP, and 6 water molecules. Okay, so let's go directly to the different characteristics. Now, take note that for fatty acid biosynthesis, it is actually occurring in the cytoplasm of the following organs. Una-una, liver, adipose tissue, central nervous system, and mammary glands. Okay, now at the same time, things to remember, you have what we call the lipogenesis. This is also another term used to define fatty acid biosynthesis. Okay, so you can actually say that lipogenesis is also fatty acid biosynthesis, where it is the synthesis of fatty acids and the esterification to glycerol to form triacylglycerols. Okay, so this usually is in the liver, that is happening for humans with with dietary carbohydrate as a major source of carbon. Now, others will call it as a de novo synthesis. Now, usually this is now, for de novo naman siya, ito yung pinagkaiba nito. So, this is two names also referring to biosynthesis. But the difference is that for lipogenesis, it's in the liver. For fatty acids that is happening inside cytosol, it's called now your de novo synthesis. Okay? Now, fatty acid synthesis is the creation of fatty acids from acetyl coenzyme A and NADPH through the action of what we call fatty acid synthase. Now, take note of the major enzyme that is responsible for fatty acid synthesis, which is the fatty acid synthase. Okay. Now, your fatty acid synthesis takes place in the cytosol and is carried out by multi-enzyme complex called FAS, or your fatty acid synthase complex. Now, the substrate, although I've already mentioned this, it's you have 8 acetyl coenzyme A, 14 NADPH, 7 ATP, and the products is 1 molecule of palmitate and 7 molecules of water. Okay. Now, some say, although later on I'll be discussing why it is somehow the opposite of beta oxidation. Okay. So let's go to the steps here. So the first one is acetyl coenzyme A is produced. Actually, hindi po ito yung mismong step. This is the preliminary step. Okay? Hindi pa ito yung step 1. This is actually the part wherein after glycolysis, kasi all of the acetyl coenzyme A are actually products of glycolysis, wherein glucose is broken down to produce pyruvate. And then it's now enters the mitochondria that will be starting point of your fatty acid synthesis. Okay? Now, once inside the mitochondria, it can actually now the citrate shuttle transport acetyl coenzyme A out of the mitochondria by combining it with oxaloacetate to forming citrate. And once citrate is in the cytoplasm, it is converted back to oxaloacetate and acetyl coenzyme A, allowing acetyl coenzyme A to be used in fatty acid synthesis. Okay? So take note, yung kanina, this is for the reaction. This is happening, shifting from cytoplasm to the mitochondria, and then here it's shifted back to your cytoplasm. Okay, so this is now the actual step. So unang-una, acetyl coenzyme A is converted to malonyl coenzyme A by acetyl coenzyme A carboxylase. So sinamarize ko na, hindi ko na siya inisa-isa kasi po yung diagram, hindi ko siya mahihiwalay-hiwalay. Then you have also malonyl coenzyme A that's shunted to FAS. Tsaka sinimplify ko na lang guys sa inyo para hindi na tayo masyadong mahirapan. And then third step is through the series of condensation, reduction, and dehydration reactions, the two carbons of malonyl coenzyme A are added to the growing fatty acyl moiety on FAS. Okay? and FAS is then recharged with another malonyl moiety, and the cycle continues. Now, each turn of the cycle results in the addition of two carbon group to the fatty acid moiety, as well as the use of one ATP and one acetyl coenzyme A and two NADPH. When the cycle has completed 7 turns, the 16 carbon fatty acid palmitate is released from FAS. Okay? So, ito ngayon yung mismong step niya. Okay? So, you have here acetyl coenzyme A, forming now your malonyl coenzyme A. And then, it's now gonna be added with coenzyme CoASH, forming now your malonyl ACP. And then, this is where ito yung sinasabi na natin na part that is happening inside the fatty acid synthase. Okay, so ito yung paulit-ulit na action. So, continuous adding fatty acyl group until you have your long chain fatty acid. Okay, so yan. So, ketoacyl ACP, hydroxyacyl ACP, enoyl, fatty acyl P, and then another set. Okay? So, paulit-ulit lang siya 7 steps until you get your palmitate. Now, the comparison of your fatty acid and degradation in terms of location, enzymes responsible for fatty acid biosynthesis are actually located in the cytoplasm of the cell, whereas those responsible for beta oxidation are in the mitochondria. Okay? Second is yung acyl groups, yung mismong carrier. So yung activated intermediates for biosynthesis is what we call acyl carrier protein or ACP. While for beta oxidation, it is coenzyme A. Okay. Now, the similarities between the two carriers both contain phosphantane group, which is made from vitamin pantothenic acid. In both cases, fatty acyl group is bound by a ester bond to the phosphantetine group. So, ito po yung mismong molecule o yung mismong compound. Yan. So, ito po yung sa fatty acid synthesis. Ito po yung sa degradation. Okay? So, almost similar sila. Ito lang sila nagkaiba. So, dun lang sila naiba. Pero it's the same vitamins. Okay, which is pantothenic acid. Okay, another one is enzyme involved. So, for biosynthesis, it's FAS synthase. While for beta oxidation, wala pong complexes na involved dito. So, yun yung pagkakaiba nila. So, may fatty acid synthase complex. Sa synthesis, sa degradation, wala po. And then for electron carriers, yung NADH and FADH2 are produced in beta oxidation, while for biosynthesis, it is NADPH. Okay? So actually, pinagkaiba lang ng dalawa is that NADH and NADPH kasi yung isa may phosphate group na naka-attach dun sa ribose. And if you're going to look at the structure here, you will see the difference. Ito lang, oh. Ito lang ang pinagkaiba ni NADH. Pag tinanggal mo siya, si NADH. Pag nilagay mo siya, it's NADPH. Parang si Superman, tinanggal yung salamin, binalik yung salamin, iba na. Okay? So, yan po. Now, any questions so far with biosynthesis, guys? Before we proceed to my regulation. Hindi. Huwag muna tayo mag-regulation. Let's go first with cholesterol synthesis muna before we go to metabolism regulation. Okay, so this one I got this from a medical doctor. Yung PowerPoint niya, sorry. So this is actually a discussion that was made by Thomas Day Spring. Now, the actual cholesterol synthesis was actually synthesized or made formal by Conrad Bloch and Fyodor Leinen. Okay. Now we all know that cholesterol is a 27 molecule which is insoluble in water. Okay. So this is actually synthesized from acetyl coenzyme A. Now take note of the structure of our cholesterol. You have there your three six carbon chain, ring chain, and then a five carbon. Okay? And then you have a hydrocarbon tail. Okay. Now they are steroid-based alcohols having hydrocarbon aliphatic side chain. This one. And then 8 carbons at the 7th position of the hydroxyl group, hydroxide here, and hydrophobicity at the hydrocarbon side chain. Sterols can be incorporated into lipid bilayer in the cytoplasmic membrane. So this is part of the cell membrane. Okay. Now, in terms of cholesterol biosynthesis, there are actually four. Okay, so take note that the liver is responsible for 15% of cholesterol synthesis, and the rest is extrahepatic, so it's outside. Then cholesterol synthesis is four states with its precursor, which is acetate. Now, we all know acetate, di ba? Kanina pa natin nakita at nag-uusapan si acetate. Okay, see, this is for ketogenesis, acetoacetate. Okay, now this is the first step is the formation of your acetyl aceto ascoenzyme A. Okay, so you have here your this is actually similar to the ketogenesis. Okay, and this is reversible. So the enzyme is the thiolase. Okay, now the second also part of the first step here. Your acetyl coenzyme A condenses to acetoacetyl CoA upon the action of cytosolic hydromethyl glutaryl synthase and becomes HMG-CoA. Yeah. And then after that, the catalytic action or rate limiting state of HMG-CoA reductase, an integral part of the smooth endoplasmic reticulum, irreversibly forms mevalonate, which is a 5 carbon, okay, intermediate with NADPH serving as the reductant. HMG-CoA reductase transcription is highly regulated by a membrane-bound transcription factor designed as sterol regulatory element binding protein 2 and different from mitochondrial uh, HMG-CoA which plays a role in ketosis. So, pinagkaiba na ni ketosis is that the enzyme here, yun yung magpapabago. Okay? So, yan. So, instead of going through with ketosis, ito po, it forms mevalonate. Okay? Using enzyme element binding protein SRABP2. Okay? Now, in stage 2, okay, so you have your mevalonate. It now undergoes phosphorylated with the help of ATP. Okay, so dinagdagan natin ng ATP, another ATP in the process, and then another ATP on this third carbon. Okay, okay. And then it can isomerize and convert it to your dimethyl allyl pyrophosphate. Okay. This is delta 3 isopentyl pyrophosphate. Ito po, before forming dimethyl pyrophosphate. Okay. So, di ba nilagyan natin ng phosphate doon. Then later on, we remove the phosphate to make it a double bond. And then we move the double bond inward. Okay. And then the third step here, the isoprenoids react with each other to form now geranyl pyrophosphate, with other isopentyl pyrophosphate yield farnesyl pyrophosphate. So ito po, geranyl to farnesyl. So you combine them together. And then squalene synthase, squalene catalyzes the condensation of two molecules of farnesene pyrophosphate, which is reducted by NADPH to make squalene. Okay, so ito yun. Okay. Now, once it's already forming the squalene, this time it's gonna be undergoing now the conversion of squalene, the linear form, to form now our squalene 2,3 epoxide. And then once it's already formed your squalene 2,3 epoxide, it will now form now cyclic rings forming the cholesterol. Now, pwede rin po from cyclase o yung squalene 2,3 epoxide, pwede siya rin mag-stigmasterol and ergosterol. Okay? So, yun yung po yung mga products niya. So, ito na po yung main output niya. Okay? Now, if you're going to simplify it without the formulas or the compound structures, ito po siya. So, acetate and acetoacetyl coenzyme A with the enzyme HMB synthase forming HMG-CoA, and then HMG-CoA reductase changing it to mevalonate. Then it forms now your isopentyl pyrophosphate. Okay, sorry guys, dapat to dimethyl PP pyrophosphate to isopentyl pyrophosphate to farnesyl pyrophosphate forming your, and then yung farnesyl naman siya, it will form your isoprenoids. Now, your farnesyl can form your squalene, forming your lanosterol, forming your cholesterol. Okay, so this is the pathway. Pasensya na po, I'll have this edited out kasi yung nung kinuha ko siya from his PowerPoint, medyo maliit masyado. I need to increase the size para makita. Now, what can we get from cholesterol? So, from cholesterol, di ba, remember, cholesterol is coming from steroid hormones, from it can form bile acids. It also forms your vitamin D. And then, yung mismong isopentyl pyrophosphate, okay, it can actually form isoprene, which is actually can form other structures as well. Then you have quinone, which is electron carrier. You have dolichols, phytol chain for chlorophyll. Then you have rubber. Then you have vitamin A, vitamin E, vitamin K, carotenoids. Okay, ito po yung actually ginagamit sa beta carotene. And then you have abscisic acid and gibberellic acid as well. Okay, so this is actually what is coming from isoprene units. Okay, so this time let's talk about metabolism regulation, lipid metabolism regulation. Okay, now we're actually gonna, in terms of metabolism for lipids, or regulation for lipids, we're going to be breaking it out based on the different organs that are going to be highly utilizing lipid inside the body. So, unang-una po is the liver, of course. This is actually the most important organ that utilizes the fats, because this is where the regulation of fatty acid metabolism rather, sorry. Okay, so unang-una, the important is that it provides supply for glucose and other system, and it regulates blood glucose by converting it to and from glycogen. So pag sobra yung glucose, glycogen ka. Pag kulang ang glucose, balik ka from glycogen. And it can produce glucose from other materials like lactate from overused muscles, and it also controls lipid metabolism. Okay, so this actually we discussed this already when we discussed carbohydrate metabolism. So this time, for lipids, if there's a surplus of fuel, liver produces fatty acid which is to create triglyceride. Now, so yan po, nag-form siya ng triglycerides, and it also transforms fatty acid to ketone bodies during fasting or starvation. Now, take note the liver lacks an enzyme that converts acetoacetate to acetyl coenzyme A, and these ketone bodies cannot be used. And as a result, the liver's ketone bodies are transported to other organs where they can be oxidized to create ATP. Okay, so here is a diagram showing it. Okay, so glucose from blood to for that is going to be shifted to the liver. It can be changed to glucose 6-phosphate, which will be used for fuel. And then pag sobra, it becomes glycogen or fatty acid. Now, your fatty acid is usually going to form now your VLDL. Yan. which is gonna be sent to the different adipose tissue pagkatapos natin kumain. Usually pag sobra-sobra, this becomes free LDL. Now, if in case during starvation or overnight fasting, pagdating ng gabi, kaya nga di ba dapat pag kumain kayo, kaya nga 'di ba dati nauso yung after 6, I'm not sure if you've heard of that after 6 dieting. So pagdating ng 6 o'clock wala ka nang kakainin. Okay, so you can have your dinner before 6 o'clock. Kasi at least yung time mo from your dinner to your breakfast, mas mahaba po yung fasting na tinatawag. Okay? So yan. So here, if you will notice, you have now your fatty acids are gonna be utilized pag hindi ka pa kumakain, and it's gonna be broken down by the liver, okay, as fuel. And then glycogen will be broken down back to glucose 6 phosphate, and it will be utilized as glucose for the blood. Okay? So ito po yung mismo cycle ng liver after meal and during starvation or after fasting. Okay? Or during fasting rather. Now, the second organ or the part of the body that also utilizes or regulated by the lipids is the adipose tissue. Okay, so usually fatty acids are mostly stored in adipose tissues in the form of triglycerides. Okay, now this is actually a fat that is created by the body. Now, fatty acids are transported from the liver as VLDL complex. Ayan. And adipose tissue absorbs the chylomicron and fatty acids. Then glycerol 3 phosphate is required to production of triglycerides in uh, adipose tissue. Now take note, di ba, remember from glycolysis, yan. You produce glycerol, no, not glycolysis. Okay, glycogenolysis, you produce that. Okay? And this in turn forms now like triglycerides, and they be forming now your glycerol. Okay? And then stored siya for storage all over the body. Okay? Now, to generate and store triglycerides, adipose cells require a constant supply of glucose. Okay? So, kanin pa more. Sweets pa more. Yan. Kaya di ba sa guys, hindi porke nagbabawas kayo ng fat sa diet ninyo, it's good na maglo-load kayo ng carbs. Make sure that you load it with the correct carbs, okay? Like vegetables, okay? Like fiber, not yung sweets, okay? Yan. Now, for muscle tissue, yan. So, take note that during rest, okay, yung energy is supplied by beta oxidation from fatty acids. And then at work, the muscle obtains energy from the degradation of its own glycogen, resulting in the building of the lactate from glycolysis. Okay, so this is actually also known as the Cori cycle. Now, alanine may be produced from protein degradation that's coming from the muscles itself, and alanine and lactate are shipped from the liver to convert it back to glucose. Yeah. Okay, so ito ginamit natin. Although in this picture that I got, it did not include alanine, pero kasama po yan. Kasi remember, alanine can alternate to form pyruvate for action of the muscle. And then, any excess alanine in the muscles and uh, lactate will be converted back by the glucose and then shipped back to the muscles. Okay? And then, actually, the only one, yeah, heart muscle prefers ketone bodies over glucose. Okay. Now another one that is also helping, that's also being regulated, is the brain. Okay, so at normal condition, the brain uses only glucose as the primary source for metabolic energy. Now, if in case it is at rest, about 60% of the free glucose is used by the brain. Okay? Now, if in case you're in a diet or in starvation, syempre naubos yung glycogen, and the amount of glucose on the brain drops sharply. Okay? Now, therefore, kung wala na pong glucose yung brain, okay, it will utilize ketone bodies as alternative energy source. And then fatty acids cannot cross the blood-brain barrier to be used by the brain as an energy source. Kaya nga minsan, di ba, pag kumakain, yung mga sinasabi ng mga matanda, ang sakit ng batok ko, lalo na magpapasko, tsaka mga may fiesta, nandiyan dyan yung mga lechon, yung mga matataba, di ba? Chicharon, yan. Sabi na, aray ko, sumasakit yung batok ko. That's because of the blood-brain barrier. Okay? Hindi yan lumalabas. Ganun din doon sa mga for those who are, I'm not saying na lahat kayo uminom For those who are also drinking, may alcohol barrier din po. Kaya yung iba po, sumasakit yung batok pag umiinom. Okay? So, ketone bodies, due to its free carboxylate group, are soluble in the blood and can enter the brain. Okay, so alternate natin is ketone bodies because of the carboxylate group in its structure. Okay. Now, let's talk about the effects of insulin and glucagon in cellular metabolism. So, unahin natin. So, we all know, we have been this, I think we discussed this also last time. Insulin is actually produced in the beta cells of the islets of Langerhans, which is found in the pancreas. Take note, may isla sa loob ng katawan natin. It is secreted from these cells in response of increased blood glucose level. Okay, so it actually lowers the concentration of glucose, causing the numbers to change the metabolism, like by storing glucose both as glycogen and triglycerides. Okay, so it activates biosynthetic process and inhibits catabolic processes and acts only on target cells that possess specific insulin receptor proteins in their plasma membranes that can be found in liver, adipose, and muscle cells. So these are the ones that has what they call the target cells. Okay. Now the blood glucose level is normally at 10 millimolar. However, a substantial meal increases the concentration of blood glucose considerably and stimulates insulin secretion. Okay. So, and then subsequent binding of insulin in the plasma membrane, insulin receptor increases the rate of transport of glucose across the membrane and into the cell. Okay, so yan po yung characteristic ni insulin. Now, the partner ni insulin is glucagon. Okay, so glucagon is secreted in the alpha cells of the islets of Langerhans. So yan o, so alpha and beta. Si alpha, si glucagon, si beta, si um, insulin. Okay? So it also affects generally the opposite effect of insulin. So it inhibits glycogen synthesis and stimulates anabolic process, which is glycogenolysis and gluconeogenesis. It also stimulates the breakdown of fats and ketogenesis. Okay, so yan yung mga trabaho ni glucagon. Now, if you're going to compare glucagon and insulin based on the different uh, pathways that we have discussed, you will notice that for cellular transport, okay, insulin increases kasi cellular transport yan eh. Insulin is the one responsible in delivering sugar inside the body. Pero si glucagon wala kasi siya lang naman yung nagbe-breakdown eh. Then for glycogen synthesis, it's increased. Then decrease siya kay glucagon. Then glycogenolysis in liver, decrease. Okay? Ito naman increase si glucagon. Now, gluconeogenesis is decrease, increase naman din. Now, for uptake of amino acid and protein synthesis, increase siya pero wala siyang effect. Same as with inhibition of proteins. Kasi remember, ito po kasi, particularly this one, is more on the conversion of proteins into usable carbohydrates. Yan. So, we're actually having conversion of this amino acids in the body to form your pyruvate, your other intermediates for glycolysis. That's why for amino acid intake and protein synthesis, it's increased in the insulin, while for inhibition of amino acid release and protein degradation, it is decreased. So wala pong epekto kay glucagon kasi nga po sugar lang siya. But for Lipolysis and ketogenesis, it is decreased both for insulin and increase in glucagon. Okay? So yan po yung mismong chart showing how how the two hormones will be affecting the metabolism. Okay? Any questions so far? Question, guys? Violent reactions?
Wala naman po.
Okay, sige. If you have no more questions, let me just end my presentation. Okay, wait lang guys ha. Ah, um, guys, we receive a memo. Wait lang ah. Before I forget, we receive a memo from from the president and also from the student affairs, VP for student affairs about the status of your survey. I know some of you may have already answered the survey that is available here. Diba, tinuro ko na sa inyo to, yung sa vaccination tracker. Parang ayaw niyo yata magsibalikan sa Yuma ka, ayaw niyo mag-FaceTime So, yes, may comment
Ano actually po ma'am, almost lahat naman na po nakapagsagot. Na-monitor naman.
na monitor na.
Yung whole class po namin.
Oo nga, pero kasi ang problema, kami naman ang inaano, medyo nababa pa daw yung update sa tracker ng mga bata. Wala pang 50% guys ang nagsumasagot. Kaya yon. So sige, okay lang. It's good that you're able to do that. Anyways. Okay. So yan, let me just go over some of the video. Wait lang ha.